Hello everyone, I'm Vishal Negi, and in my previous lecture I talked about Introduction to Oncology, in which I covered study of cancer, various characteristic features of cancer cells, and types of cancer. In continuation to this, today I'm going to talk about molecular basis of cancer. So first of all, I would like to cover how cancer develops. Previously we saw that mutations in genes, particularly genes which encode cell cycle regulatory proteins, both positive or negative regulators, causes development and progression of cancer. So fundamental thing is mutation causes cancer. But we need to keep it in mind that single mutations usually do not lead to cancer or do not causes cancer. Cancer is basically developed because of multi-step process and multiple mechanisms must fail before a cell becomes cancerous. That means most cancer arise as cells acquires a series of mutations. So this is very important, a series of mutations. So cancer will arise because of series of mutations. So for example, mutation in cell cycle inhibitors may lead to benign tumor. Okay, and benign tumor is basically a mass of cells that divide too much but don't have the potential to invade other tissues. So in my previous lecture I talked about the unlimited cell division potential of cancerous cells. So when normal cells become benign tumor, they acquires the capacity or property to divide for unlimited period or for unlimited number of times. However, they do not have the potential to invade other tissues. So this benign tumor becomes malignant tumor by acquiring some more mutations. So in order to convert a benign tumor into a malignant tumor, cells need to acquire more mutations. And because of that, now besides having the property to divide for unlimited number of times, it also acquires the property of metastasis, through which it can break down from the actual site of tumor formation and spread into other parts of the body. This is how a cell becomes malignant. So mutation is the fundamental process that is required to convert a normal cell into a cancerous cells, but it requires a series of mutation and not just a single mutation. Now let us talk about molecular basis of cancer. So as I previously mentioned, regulatory proteins are of two types, positive regulators and negative regulators. Positive regulators are the proteins which promote cell division, and negative regulators are the proteins which inhibits cell division or prohibit the cell division when the conditions are not right or favorable. So any mutations in positive or negative regulators can lead to cancer. Now, so these mutations can either make positive regulator overactivated and negative regulator inactivated. So what does that mean? So if you remember the role of positive regulator, it is to promote the cell division. And if it is overactivated, then it will promote the cell division in an uncontrolled way and cell will keep on dividing without any check or inhibition. On the other hand, if negative regulator is inactivated, what will happen? So the role of negative regulator is to inhibit the cell division, and if the proteins which are responsible to inhibit the cell division when conditions are not right, then what will happen? Inactivation in those proteins will result in uncontrolled cell division even if the conditions are not right. So when the normal cell was supposed to stop the cell division because of unfavorable conditions, it will keep on dividing, and that will lead to the formation of cancer. So overactivation in positive regulators or inactivation in negative regulators can lead to the formation of cancer. So positive regulators, when they become overactivated, we call them oncogenic because they promote cancer. So overactivation of positive regulators make them oncogenic. And negative regulators, the example is tumor suppressor. So when it is inactivated, when tumor suppressors are inactivated, then it leads to cancer. The normal genes of positive regulators. So when I say normal genes, I mean not mutated forms. So there is no mutation in normal gene of positive regulators. At that point, we call them proto-oncogenes. And overactivation of proto-oncogenes. Overactivation means any mutation that can lead to overactivation converts them into another form which is called oncogenes. So mutations in proto-oncogene that results in increased activity of positive regulators convert those proto-oncogenes into oncogenes. So proto-oncogenes are basically the genes which encodes for positive regulators, and they are normal genes, means there is no mutation. But when mutation occurs in proto-oncogenes, which codes for positive regulators, and if that mutation results in increased activity of oncogene, then it increased activity of proto-oncogene, then it will convert it into oncogene. So basically proto-oncogenes encodes for regulatory proteins or positive regulators. An example is RAS, which is a G protein, and the role of RAS, a positive regulator, is to transmit growth factor signals when there are growth factors available. So I always talk about favorable and unfavorable environment for cell division. So a cell will require growth factors to divide, and when growth factors are there, then RAS protein transmit growth factor signals so that cell can divide. And if there is a mutation in proto-oncogene or RAS-A protein, then what will happen? Mutation may cause the protein to transmit signals even when no growth factor is around. So no growth factor means unfavorable conditions. So conditions are not right for the cell to divide. It requires growth factor. So even when there is no growth factor, mutated form of proto-oncogene, which is now an oncogene, will transmit the signal for the cell to divide, and it will lead to uncontrolled cell division. So the proto-oncogenes encodes for positive regulators, and positive regulators are in two forms, inactive form and active form. So we can call it on and off form. So when it is in on form, it is in active form, and when it is in off form, it is in inactive form. So these two forms are basically distinguished by the property of RAS protein to bind with GDP or GTP. So when it is bound to GDP, it is in inactive form, and when RAS is bound to GTP, it is in active form. So basically, the protein has ability to switch between active and inactive form. However, mutation in the proto-oncogene that encodes for RAS can change the structure of RAS in such a way that it can no longer switch to its inactive form. What does that mean? It means the RAS protein, which basically can be converted into inactive or active form because of its ability to bind to GDP or GTP, it will no longer be able to convert into inactive form because there is a structural change that will no longer allow GDP to bound to RAS protein so that it can be converted into inactive form. So now RAS protein has some structural change that will allow only GTP to bind to RAS protein, and that will results in permanently on state of RAS protein. And permanent on state of RAS protein means the positive regulator is mutated or overactivated, and now the cell division will keep on going even if the conditions are not favorable. So cell division is basically now happening in an uncontrolled way. That is one of the hallmark of cancer. Now let us understand the concept of oncogene by the help of this diagram. So this is, let's say this is a normal cell, and in case of normal RAS, that is encoded by proto-oncogene, which is not mutated. So in case of normal RAS, what happens? So when, let's say when the conditions are favorable, so there are growth factors in the extracellular environment which a cell require in order to divide and proliferate. So when there are growth factors, the growth factor receptor, this is shown in the blue, of a cell will recognize the growth factor. So once growth factor binds to the growth factor receptors, it results in dimerization of growth factor receptor. So the dimerization of growth factor receptor results in the activation of RAS protein. And what does activation means? It will have GTP bound to it. So now RAS is bound to GTP, that is an on state of RAS or active state of RAS. So this activation of RAS results in a series of signal transduction cascade that will give cell a signal to divide and proliferate. So this will result in cell division and proliferation. In case of lack of growth factors, when growth factors are not around, the growth factor receptor will not dimerize. As a result, it will not give any signal to the inactive form of RAS, that is RAS GDP, to become active. So in case of no growth factor, there is no dimerization. As a result, RAS is in the inactive form. That means RAS is bound to GDP, and that is off stage. So as a result, there is no signal transduction cascade to promote cell division or proliferation. So this is the case of normal RAS, when RAS is encoded by proto-oncogene. But what will happen if there is a mutation in proto-oncogene which encodes for RAS? So that will encode for oncogenic RAS. And as I mentioned, oncogenic RAS may have some structural change that will keep it in permanent on position. So RAS will always with GTP, because now structure of RAS is changed in such a way that it can no longer bind to GTP. It can only bind to GTP. So even though there is no growth factor, even though growth factor receptors are not bound to growth factors, and even though growth factors receptors are not in the dimeric state, RAS does not need any signal from the receptors. RAS will always be in the active form because it is bind to GTP, and there is no possibility that the active form of RAS can be converted into the inactive form. As a result, RAS is always active and always in the on position, and that is why it will initiate a signal transduction cascade. As a result, cell will keep on dividing permanently in an uncontrolled way, and that is one of the hallmark of cancer. In contrast to oncogenes, tumor suppressors are the negative regulators of cell cycle. That means they inhibit cell cycle progression when the conditions are not favorable. So tumor suppressors basically are the negative regulators which prevent the formation of cancerous tumors when they are working correctly, and tumors may form when they mutate, so they no longer work. So the bottom line is, we want tumor suppressors to be in the active form, and any inactivation in tumor suppressor will result in the formation of cancer. So negative regulators of the cell cycle may be less active or even non-functional in case of cancer cells. And the example of tumor suppressor protein is p53 protein, which plays a key role in the cellular response to DNA damage. So whenever there is any DNA damage, p53 acts. So p53 acts primarily at the G1 checkpoint, where it blocks cell cycle progression in response to damaged DNA and other unfavorable conditions. So let us see this in a diagram. So in this diagram, you can see that this is a normal p53 protein, and whenever there is a DNA damage, p53 binds to the DNA and activates the CDK inhibitor. So this CDK inhibitor binds to CDK and inhibits CDK and G1S cyclin complex. As a result, there is no cell cycle progression. So cell halts at the G1 checkpoint. DNA repair is activated, and if it is repaired, cell will resume the cell cycle. If it cannot be repaired, if repair is impossible, then apoptosis will be triggered, and apoptosis will lead to the death of the cell. So basically the fundamental point is p53 helps a cell to inhibit the cell division and do not let the cell pass the damaged DNA to the daughter cells. However, if p53 is non-functional, means it will no longer be able to inhibit the cell cycle. So even if there is a DNA damage, p53 will no longer will be able to bind to the damaged DNA. As a result, it will not be able to activate CDK inhibitor, and in the absence of CDK inhibitor, CDK and G1S cyclin complex will be active. As a result, cell cycle will progress. So what will happen? The damaged DNA or the mutated DNA will pass on to the daughter cells. So now we have seen one example of oncogene, which is RAS, and one example of negative regulator or tumor suppressor, which is p53. So now it is important to notice that one is a positive regulator and other is a negative regulator. So the role of positive regulator is to promote cell division, and the role of negative regulator is to inhibit the cell division. And what kind of mutation leads to cancer? In case of positive regulator, it is overactivation of positive regulator, and in case of negative regulator, it is inhibition of negative regulator. So let's say we have two normal copies of RAS. So two normal copies of RAS, in case of normal cell growth, the daughter cell will have normal copies again, and normal cell growth will happen. Okay, so there is no cancerous growth, there is a normal cell growth. However, out of the two copies, even if one copy of RAS is mutated, that means this one copy is overactivated. And when this is overactivated, despite having one normal copy, this mutated copy will lead to the formation of cancer, because it will be in the permanently on state. As a result, cell will get the signal of cell division and proliferation even if the growth factors are not available. As a result, cell will keep on dividing. So the bottom line is, even one mutation, even a mutation in one copy is good enough to promote cancer growth. However, in case of p53, which is a negative regulator, so right now this cell has two normal copies of p53. Upon cell division, it has two normal copies again, that will leads to the normal cell growth. Let's say if one of the daughter cell has one mutated copy. So now one of the tumor suppressor p53 is mutated and the other is normal. So upon cell division, one cell will have one normal and one abnormal copy. So even though one copy is normal, it will lead to normal cell growth because its role is to inhibit the cell division. So even though the one of the copy is normal, it will serve the purpose. But if both the copies are abnormal or both the p53 copies are mutated, then they will no longer be able to bind to the damaged DNA and they will not be able to repair it. As a result, cell will be in permanently on state, and that will cause the formation of tumor, right? So in case of negative regulators, we need both the copies to be mutated, but on the other hand, in case of positive regulator, even one copy mutation in one copy is good enough to promote the cell division. So that's all for today's lecture, and now it is the time for an assignment. So I have three questions for your assignment. First is, what is an oncogene and how does oncogene be related to the formation of cancer? What is a tumor suppressor gene and how do it is related to the development of cancer? Explain why mutation in one copy of proto-oncogene is good enough to promote tumor formation while a mutation in both the copies of tumor suppressor is required for the same. So I will post these questions in Google Classroom, and please solve it over there so that I have the record of your assignment completion, based on which I can grade you and I can give you attendance. Thank you for listening to me and take care. Good luck.